people who didn't come to the advanced lecture might want to look at the first half of it. The first half of it sort of expands upon this, what does this diagram mean? So everybody knows how to draw this diagram, one pattern direction, and then a whole bunch of local directions that are consistent with it across space. So what does that actually mean? So go to the first half of the advanced Friday lecture, and you can sort of see that sort of expanded out into its full glory. Okay. So what's the topic for the main lecture? So the topic is attention. So you've probably read a lot about attention. Attention is very, very important for deciding what to look at next, or you've got a complicated mess of a scene, and you want to pay attention to a particular part of it, and at the same time ignore other parts of it. So it's just not attention to good things, it's also suppression of response to things you don't want. So when you think of, if you go to a psychology textbook and read about attention, where is attention? So if you go to a psychology textbook and say, let's look in a monkey brain and try to figure out where the attention is. So there's the central sulcus right there. Here's the STS going up like that. Here's the interparietal sulcus. Here's the lunate sulcus, and here's the principal sulcus and the arcuate. So there's a monkey brain. So, where is attention? Well, if you read in a textbook, it's somewhere in parietal cortex. There's some attention there, and then maybe there's some attention in frontal cortex. That's usually where you think of the focus of where is attention in the brain. And what we're going to talk about today is that's completely wrong. Attention isn't in one place. It's pretty much, attention is kind of a mechanism of competition between and control of stimuli that's essentially present everywhere. Virtually every neuron in the brain that has some response to a visual object has attention in it. So there's some attention signals in V1. There's attentional signals in V2. There's attention signals in infratemporal cortex. There's attentional signals in parietal cortex. There's attentional signals in a bunch of places in frontal cortex, pretty much all over the place. So I'm going to talk about one experiment in detail. This experiment has been, again, like a lot of things that we talk about in this course, like somebody does this one experiment and it's very influential, and then people do it again 500 times. So this experiment has been redone in different versions like 500 times. I just want to go through the first version of it to sort of give you some idea. Now, where were these guys recording? So here's V1 right there. So V2 is kind of stuck in this sulcus. So you can't see V2 from the surface on a monkey brain. It actually sort of is actually sort of inside the sulcus. And there's another area called V3, which I talked about a couple weeks ago. That's also inside the sulcus. And then when the sulcus comes out, there's an area of V4. And so why have people done a lot of work on V4? Part of the reason is it's easy to get to. It's not inside a sulcus, and so if you're trying to record from a neuron there, it's right on the surface, easier to get to. So that's one of the reasons people have done a lot of work there. So let me just label these. So traditional theory, parietal, frontal. Okay, so V4 isn't parietal or frontal, but there's big attention effects there. So let's look at them. And so what is this experiment? So this experiment was Moran and Desimone. People did experiments before this that were like this, but this was one that, what is that? 1986 maybe? I think it was 86, something like that. 1985 maybe? How about 5? So this was an experiment done in a monkey, in a macaque monkey, in area V4. And it was looking at attention. Now, one thing, when you talk about experiments in monkeys, it's worth sort of talking about, like, what do macaque monkeys do all day long? Now, all monkeys aren't like this, but macaque monkeys are. So this was done in macaque monkeys. So what do macaque monkeys do? Well, if you've ever seen, you can go to the zoo, and there's a number of different species of macaque monkeys. Baboons are relatively similar. And so what do they do all day long? So here's a macaque monkey. And here's another macaque monkey. And so here's this guy looking that way, this guy's looking that way. So if you go to the zoo and just look at a bunch of macaque monkeys, what are they doing? Well, they're all looking off in different directions. Like this guy's studiously looking that way, and this guy's studiously looking that way. But what are they actually doing? So it turns out in macaque monkey packs, it's usually a polygynous pack, so it has like a dominant male and then some subdominant males that are off to the side, and so those guys have to sneak sex so that the dominant male can't see what's going on. So what do these guys do all day long? They studiously don't look at each other. And it turns out in macaque monkeys, looking straight at another macaque monkey is a big threat. Now, in humans, it can be a threat, but it's not uncommon that another human will look you right in the eye. It's not a problem. In fact, if they don't look you right in the eyes, you might think, there's something wrong with them. They're not quite right. But in macaque monkeys, this is a big threat to look somebody straight in the eyes. And so what do macaque monkeys do? They spend their entire day attending to things that are different from the ones they're looking at. So basically, these are pre-adapted for these experiments. So they spend their entire day, peripheral attention all day. So if you go to the zoo and look at a macaque monkey in the eye, what will happen? They'll start they might threaten you, like, stop looking at me, it's like, that's an example of where it's very clear that humans are not aware of the fact that the social signals in macaque monkeys are different than humans. They'll just they'll sort of unintentionally threaten the macaque monkey, and then the macaque monkey will get excited and maybe slightly charge the person looking at him. So they spend all day essentially. So this guy if this guy's the dominant male here, this guy's carefully paying attention to what that guy's doing, looking somewhere else all day. So it's actually sort of pre-adapted to doing peripheral attention experiments, which is what we're going to talk about. But the other thing that macaque monkeys are pre-adapted, so these are pre-adaptations for attention experiments. They like small rewards. So some monkeys, like say owl monkeys, they eat fruit, and fruit is kind of like a big, you got a big juicy fruit, that's a big reward. Macaque monkeys will, here's an example, I always give this example. Here's something a macaque monkey will eat. It will go someplace, find a blade of grass, and then there's a little tiny white root under there. Pull that single blade of grass out and just eat the white root. and they'll do that thousands of times in the daytime. So they're well set up for being trained with small rewards. Whereas if you take an owl monkey is expecting like, a whole big juicy fruit. You give it a little thing like a blade of grass, it's going to go, yeah, I don't care about that. It's not. So it's much harder to train them. It's easier to train these guys because they're kind of used to this. So importantly, what is our definition of attention? So what's the operational definition? Because of attention. So so what does the experiment consist of? So the experiment consists of basically first having the monkey fixate. So what does that mean? That means the monkey has to look at a particular point like that X. And so why do you need the monkey to fixate? Well, you can think of the receptive field as attached to the retina. So if you have a receptive field, say a receptive field in the lower, so this is say a V4 receptive field. So if you have a receptive field in the lower field, the problem is if the monkey looks around, the receptive field is going to move with the retina. And so like wherever the monkey is looking, say here's the receptive field, that receptive field is going to move all around, and so then you won't know what's actually in the receptive field. So the first thing you have to do is train the animal to fixate. But then the second thing you need to do is to train the animal to selectively attend to something. And what is the kind of thing that people have used? Well, they've used something like, let's put like a little red bar in there, and let's put a blue bar in there. So you got two stimuli in there, and the operational definition of attention is respond to small changes in one of those and not the other. So that's the operational definition. So respond only to small changes in one of the stimuli. So obviously we don't really know what the animal is attending to, and so we just operationally define this as I slightly dim the blue bar, and if that's the one they're supposed to be attending to, then they're supposed to press the button. If I slightly dim the red bar, they're just supposed to ignore that. And so after the animal is doing this repeatedly, it's responding to these slight dims of the blue bar and completely ignoring the slight dims of the red bar. And you could change the task around so that the animal now has to pay attention to the red bar and ignore the slight dims in the blue bar. So it's important to sort of dig down into the paper and just figure out what do we mean by attention. So it's the ability to ignore slight changes in one stimulus while paying attention and responding to. That's how we essentially define it, by the response to the one that the experimenter has determined is the one that you're supposed to be attending to. So that's the way all these experiments work. Now, how do you get a monkey to do this? I always spend a little time talking about monkeys and undergraduates, and so if you ask it, how would you do this with an undergraduate? You would say, press the button when the blue bar changes and ignore the red bar, and you're done. So that's good. And most undergraduate, depending on what they had last night, most undergraduates would do a pretty good job of that. Now, how do you do that with a monkey? How do you tell a monkey to only press the button when you've got a vertical blue bar? Hard problem. This could take several months. And so these experiments are extremely time-consuming. So what you have to do is you first have to get the animal to fixate. And so how do you get the animal to fixate? Well, you're monitoring the eye position of the animal, and the animal's looking all around. And then you want it to fixate. So how do you? Well, you cut up a banana into 1,000 pieces, and anytime you look at the monkey, and anytime the monkey looks anywhere close to the fixation point, you give him a little bit of banana. Now that could take like a month to train the animal to do that. But after the animal has the concept that banana is associated with looking at the fixation point, they'll look right at the fixation point. So that's the first thing. But then, how do you get them to actually respond? Well, you put a little lever in there. So they got a little lever like this, and here's a little monkey hand. And so the monkey looks at that lever and maybe presses it a couple times, nothing happens. And so it kind of ignores the lever. And so then you cut up the banana again, and anytime the monkey's hand gets anywhere close to the lever, you give it a little banana. So finally it figures out, it's the lever. And so then you have to press the lever. Okay, so once the animal's figured out to press the lever, now you have to let go of the lever, because it'll go like, yeah, right, that's the banana thing. So months later, once you've got them to press the lever, and then they realize that when they press the lever, they sometimes get a reward if they were responding to the blue one, and they don't get a reward when they respond to the red one. So months later, they figured it out. Once they finally figured it out, they're faster than undergraduates, about twice as fast. And the reason is their brains are smaller, and so all the conduction times are shorter. And so once they've learned this, they're just like machines. They're just looking at it, ding, 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 just like really, really going fast at it. But have respect for how long it takes to actually train the animals to do this. It's really quite a time-consuming operation. And so what was found out here? So now the idea is we have this animal trained, and it can attend to one or another stimulus, and just attend to that one. Let's just look at how those stimuli interact with each other, but also how do they compete with each other. And so we have a number of different conditions to look at. And so first condition, so in all cases, remember, we're only presenting the stimulus in the receptive field. So say here's the V4 receptive field in the lower field. So if we present the stimulus somewhere else, the V4 neuron's not going to care. So the first thing you have to do is figure out where the record from a neuron, figure out where the receptive field is, and then arrange the computer program to put the stimuli in the receptive field. And so the first thing we can do is, now the other thing is that the receptive field itself, this particular neuron may have some selectivity for the stimulus. And so say it likes a vertical green bar. So we'll call this condition passive good. So we can have the animal not do an attention experiment, just fixate and just ignore the stimulus basically, and then we can test different stimuli. And what we find is that some stimuli, attention aside, are better than other ones. And so here's another test, and we could just test with a red bar, and that's not a very good stimulus for this V4 neuron, so this is passive bad. So that's passive bad. And so we could look at the responses. And so if you look at the responses, we'll present the stimulus here. So let me put now there's a little bit of a delay. It's, like I said, it's shorter in macaque monkeys than it is in undergraduates, but maybe like about a 40 millisecond delay before the response comes up. And true to my word, all the graphs in neuroscience look like that. So what is that? That's the response rate, of V4 neuron. And generally it takes like about something like 40 milliseconds for the neuron to start responding. If it was an undergraduate, it would take more like 80 milliseconds. But so there's about 40 millisecond delay, and then there'll be a response. Now, if we look at what happens to the same neuron if we present a stimulus that the neuron doesn't like, because the neuron has its own selectivity, like it likes vertical green bars and it doesn't like horizontal bars. So what will happen there is we'll get a less of a response, something like that. So it's selective for vertical green bars. So now we can do our attention manipulation, and in this first version of the paper, there were only 4 conditions. I'll add a 5th to sort of fill it out, which people have done repeatedly in many later experiments. But the first one is put both stimuli in there now. So both attend good. So now we have that same fixation point and the same receptive field, and now we've put both the green bar and the red horizontal bar in there at the same time, and now we're having the animal attend good, and so we'll indicate that by here's a little spotlight of attention. Now, whether it actually is a spotlight like that is a good question, but this is just indicating we're attending to the good one, the passive good one. So now what happens, so if you look at the original paper, this turned out not to be true in general, but if you look at the original paper, if we plot the first response there, what they saw was that there was even a slightly better response. It wasn't a big effect, but there was like a little boost in the response, a little boost. So because people were initially expecting attention to be positive, like I tend to that, and so I should boost it. Turns out it's a little more complicated than that. But so that's both attend good, and then we could alternatively do both attend bad. And so that would be the same fixation point, exactly the same stimulus. The only difference is that the animal now has to ignore changes in the good stimulus and only pay attention to changes in the bad stimulus. And so so that both stimuli are in there, but now we've got our spotlight of attention, which is on the bad stimulus. And if we look at that one, what happens is you get a suppression of the response. So the good stimulus is still in there. But the neuron is now quite a bit suppressed compared to this condition where both stimuli were in there and the animal was just responding to changes in the good stimulus. So that was the initial. So the suppression in the first paper was a lot bigger than the expansion, but so that was the way it was first sort of visualized, is that attention is capable of boosting up the response, and it's also especially capable of suppressing the response. But in hundreds of experiments that were done in the succeeding years after this experiment, among other things they did was passive both. So that's a condition that we should have tried. We did passive good, passive bad, but we didn't do passive both. So what this is same fixation point, and now we have the animal not do any task, just ignore what's going on in there, doesn't matter, and just put both stimuli in there at the same time. And now we can compare that. What does this look like compared to, say, passive good? And that resulted in the discovery that there's competition between the stimuli. So if I put if I put passive good, so this guy's passive good. So I put passive good there, and look at what happens when I present both stimuli. What I find is that there's actually suppression. And so basically, that gave people the understanding that you have, stimulus competition. And this is a big time problem. So how many times are you in the world and there's a completely black background and there's a small horizontal red bar? Quite an uncommon sort of situation. What's the normal situation in the world? There's junk all over the place. There's people in different places. There's leaves and branches and tables and chairs, and it's just like a mess of different objects. So, and what happens in that condition? Well, it turns out it suppresses the response. So there's competition between different stimuli. You can sort of think of this in a very crude way as you just tend to have lateral inhibition in the cortex. And so if one part of the cortex becomes active, it tends to kind of like dampen down areas that are around it. And so basically there's competition between stimuli that results in the suppression of the response to the stimulus when you compare it to that stimulus by itself against a black background like neuroscientists like to use. And so why do they do that? Well, you get a really big response. That's the way to get the best response, is have nothing else competing. But what has the modern understanding of attention, sort of come to? So we have this, we have this problem of stimulus competition. So as soon as we get another stimulus in the field, it's going to reduce the response to a stimulus, even a stimulus like this passive good one that the cell likes. So this cell is looking for vertical green bars. If we just add a red bar to it, we get less of a response, even though the vertical green bar is there. So we have stimulus competition. And what is attention? So attention is basically, the way to think of it is, rescuing, attended stimulus response from stimulus competition. So that's kind of really what it does. And so it's actually pretty hard to get to essentially have attention give you a response that's better than a stimulus against a black background. So another way of thinking of that is you could think of it as, as if other stim's not there. So that's kind of, the bottom line about what attention in all these different areas really does. You look out at a cluttered world, a bunch of junk there. You've got neurons that are sensitive to different aspects of that, say like, vertical green versus horizontal red. And what does attention do? Attention essentially, that's not bad, rescues, yeah. So what does attention do? Attention basically gets you back to a situation where you've got like just a green bar on a black background. So it kind of suppresses the effects of all those other stimuli out there on the one that you're attending to. It generally doesn't tend to boost it above what it would be if you weren't attending to it. It just kind of like rescues it and just sort of allows the selectivity of the neuron to be expressed. So that's really what attention does. So it kind of actively damages some of the other stimuli that you're not attending to. But this mechanism is present in all different areas. You can see it in MT or MST. You can see it in infratemporal cortex, where cells are sensitive to objects. They have big receptive fields. They're sensitive to objects. You can see it in parietal cortex. You can see it on some of the medial visual areas. You can see it all over the place. And if you think about it, it makes sense it should be in more places because all these different areas are selecting different things. Like some of the areas are more sensitive to motion, or maybe some of the areas are more sensitive to rotations and dilations, or some of the areas are more sensitive to color and they don't care about motions. So it makes sense that you should have this kind of competitive mechanism spread throughout the entire brain. So there isn't really any focus, there's no one area that does attention, they all do it. And they're all doing different kinds of attention. Some of them are sort of like attending to color versus attending to the location, or some of them are attending to motion. So it's all over the place. So that's the Moran and Desimone experiment with a more modern interpretation of it. But in the process of doing these experiments, one of the things you might think about when this display first comes up, how does the animal figure out, like, which one to attend to? It's kind of a little confusing because the animal doesn't know where these stimuli are going to be exactly. And so one of the things it has to do is look at it, figure out, I don't want the green one. I'm only paying attention to the red one. How does that work? And so people have actually looked at the time course of some of these things, in more detail. So let me just say one thing, before I talk about that, and just talk about the spotlight. So I drew this thing out as if it's relevant to the discussion about different areas having different kinds of selectivities. So, is there actually, is this the way it's actually working? In other words, is there a little spotlight that just hones in on the red bar and ignores the green bar? So one of the problems is that how would you distinguish that from something that just looks for green bars wherever they are, or looks for red bars wherever they are? So is it actually, a spatial spotlight? So how would you try to experiment with that? Well, you could try. Why don't we put the bars in the same place? They can't be in exactly the same place because they would completely overlap each other. But say we just put them on top of each other. That might be one way to test whether or not it's spatial. But it could be something else. It could be just attention to a feature. It could be like orientation. Like I'm just going to look for a vertical bar wherever it is. Or maybe it's color. That was what the experimenters were using, but the animal could also just use the orientation because they kept the orientation constant. So it's not clear that it's a spatial spotlight. It could be also some kind of spotlight where you just say, I'm just looking for red things no matter where they are. It's drawn out in the paper as if it was a spotlight, but there's no hard evidence that it actually is that way. Okay, so that was, whether it is a spotlight. But the second thing that I introduced before I interrupted myself was, how does brain decide which to suppress? That's actually a problem. And so what we can do is just blow it up, draw all the graphs on the same page. So we'll start off with the response to the good one. So this is passive good. And if we look at passive attend both, it's about the same. That was the kind of conclusion from here. It pretty much rescues the response. So this guy is, this dashed line is, both attend good. So that was both attend good. So that's the rescue. So it rescues the response that you would get against a black background if the other stimulus wasn't there. So now let's plot on here passive bad. So here's passive bad. And then our key plot is both attend bad. So what's going on in this case? Because we got the good stimulus in there, and somehow the animal has to figure out which is the good stimulus, which is the bad stimulus. Don't pay attention to the good stimulus. Just pay attention to the bad one that the cell doesn't like, which is like the red stimulus. But the other stimulus is actually in the receptive field. And so what goes on? So if we draw, so here's 0, and then here's about 40 milliseconds, and here's about, say, 80 milliseconds. So this is a short period of time. It's a 20th of a second. So what happens if we plot out both attend bad? What we find is that the initial parts of the curve look like you were pretty much seeing the good stimulus. And then the response is suppressed. So this is, I exaggerate a little bit, but, for effect, both attend bad. So what's going on? Well, it looks like in this part of the thing, that's just the delay. We have to go through, V1, DLGN, V1, V2, V4. We got some retinal delay too. Retinal, DLGN. So we got to go through a bunch of stages until we get up to V4. So that's our initial delay. But then what happens? It looks like the good stimulus actually just flies through the system. And so for another about 40 milliseconds, it looks like we get a first pass. So the stimulus actually, this good stimulus actually, because this neuron is sensitive to vertical green bars, responds strongly to that vertical green bar. And then what happens is somehow in this stage here, we get some kind of, this could be, top down, could be like a higher area, but it could also be within the area. So the area itself might figure out, oh yeah, I don't want that green one anymore, so let's stomp it out. So are we understanding how this whole thing works? No, not really. There's 40 areas, and they're all connected back and forth to each other, and there's signals coming in with various delays between these different things. So we don't really know. This is only just a kind of like a glimpse into some complicated negotiation between all these different cortical areas. But it gives you a feeling that, the system, the system needs to figure out which one am I going to suppress. And, it can't tell in advance where that object is going to be. So you can move these things around in the receptive field, and the animal has to figure out, oh yeah, that's the red one. I don't want to pay attention to that one. Let's turn that one off. Now, whether that's done, within the area, or is it done by sending it onto a higher area, and the higher area figures out, oh, that's the green one, and then sends back, this data can't show us. You'd have to record from all the areas at the same time, which is pretty much impossible. But this gives you some idea of the complexity of all these attentional things, which are happening in every area for every stimulus that you look at. And so the bad news is that you can't just go to parietal cortex and parietal cortex is going to be doing your attention for you, because it's happening across the entire brain. And it's different in every area because of the stimulus, the features that a given area are picking up are different, and so the stimulus competition is going to be different in different areas. So it's a complicated mess, but it's how the system works, and how the system works so well given very slow operating parts. So the operating parts are only firing like 30 spikes a second or something like that. So they don't have a big range of responses, 0 to 30 spikes a second. Say they were going 50 spikes a second. If they were going 50 spikes a second, that's 20 milliseconds. So like spike, 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 so you don't have that many spikes to play with here, because already attention is kicked in by 80 milliseconds. At best, you could have done like 4 or 5 spikes. So you can see how efficient the system is with relatively small number of impulses in order to be able to do all these interactions. So now is there any questions about the attention experiment that has been done over and over again in all different areas? It was originally done in V4, but they've done similar experiments in many other areas by now. Yeah.
So just to confirm, so the passive good and both attempts are nice. The passive good is the dash.
Yeah. So these are basically the same. So we could say that the passive, the both. I made the dash to both attend good, but it doesn't matter. They're basically indistinguishable. So the idea here is that the attention will rescue that stimulus competition and bring us back to as if we just had the passive good in the receptive field. So any other attention questions? So like I said, who knows what attention really is? We just have this operational definition of attention, which is the stimulus that the animal responds to. It's essentially all we're saying. So we don't know what it's actually thinking. Maybe it's attending to both, and it's just responding to only one. So, and you have the same problem with people. You don't know what they're thinking. I mean, there have been some experiments done showing that you can attend to 2 objects. So it's possible to attend to an object over here and another object over here, and you can show that the attention is less in between. So that suggests that you do have some kind of spatial attention, and it can at least do 2 objects, maybe 3. And so it can basically ignore objects that are in between 2 objects and pay attention to two at once. This would be good for monkeys too, because there might be some angry subdominant monkey ready to attack the dominant monkey. You got to pay attention to both of those. Okay, so in the last 5 minutes, I'll just talk about one more experiment. It's not explicitly related to attention, but it's another kind of experiment that has often been used for attention type experiments, and it's a different kind of experiment called match to sample, but it's a very commonly done experiment. And so we just sort of go over an example of this. And so this was a cross-modal. So it was a cross-modal match to sample. And I always talk about this experiment because just to sort of emphasize how hard it is to train monkeys. So basically, there was a visual stimulus and a somatosensory stimulus. And so the somatosensory stimulus was like a little plate with grooves on it. And so and the animal couldn't see it, it just touched it. So here's the, the animal just touching that somatosensory stimulus. And that somatosensory stimulus was the sample. So matched to sample. So here's the sample. And then what happened is, we had all the tests. And so the tests would then be little visual orientations. So here's, we might put that one up. That's obviously wrong. Put this one up, and then put that one up. And so the animal basically has to respond. They did a version of it with a visual one too, so, respond. So the animal is supposed to respond to that. You could also do a visual version of this experiment by just putting up a vertical grading like that, and then doing the same experiment. But the one we're talking about, the main one we're talking about here, was let's have a somatosensory orientation, and then the animal responds when it gets a match. So that's a match to sample. So this is, the test. And so they trained the monkey to do this, and the monkey was doing it. It took like 4 months. So they trained the monkey to do this experiment. First, this thing is like, what the hell is that thing? Touching it, it's moving around, it's kind of complicated, but they figured out how to do this. And they're recording in V4 again. And they're looking at the monkey and they say, like, I just have a feeling that monkey is not paying attention to the stimuli. The animal was doing the task perfectly well, but it just was like, I have a feeling like it's just not bothering to look at the stimuli. And so they turned off the monitor with the visual stimuli on it, and the animal was still at 100%. And so then they said, well, what's going on? How come he can do the task when he can't see the sample? And it turned out when they programmed, so the way these experiments work is usually the animal is kept a little thirsty, and then the animal gets a little drop of juice when it does the right thing. And so the animal will do the experiment for an hour until it's no longer thirsty, and then it's just, it will look away like, I'm done, take me out of here, I'm not paying attention. And so you got about an hour to do the experiment each time. So the animal is just trying to get juice. And so the animal figured out there's a little click right before the right one. So the animal doesn't know, it's just like, it just tries to figure out, like, how do I get the juice? And so when they programmed the experiment, they accidentally, the juice dispenser made a little click right before the stimulus came up, and the animal picked that up, and it was, it was doing 100%. It was saying, like, I've got to fixate, there's these things up there, just ignore them, and then just listen for the click. And so they fixed that problem, and of course the animal was going like, dang, now what? This click isn't working anymore. And so then it had to actually learn, so the animal was doing everything. It was touching this, it was fixating, looking at these things, waiting for the click. So after another 3 months, the animal figured out how to do the real task, And interestingly, when you actually looked at the response, it was very interesting because what happened, so what the experiment was looking at is, can we see any effect of what the animal was looking at on the responses to these guys? So if you look at the experiment, what it looks like is when you first present the somatosensory stimulus, there's actually a little bit of a response in V4. And then when you present the test samples, let me just line it up with this. Yeah, so what happens is when you first present this thing, there's actually a little bit of a response, elevated response. And then when these things come down the pike, this is a V4 neuron, and so it likes visual stimuli. Then you get big responses to these. And that's what the experiment was reporting on. So this is, the paper reported on, modulations of this, like attentional modulations, this visual response. But interestingly, there was this little elevation of response here. And what is that? That's probably some kind of like visual imagery. Because what's going on is the animal sees this thing. This is a visual area, and there's a little elevation of response of the neuron. So this might be some kind of, imagery where it's slightly activating neurons. There's no stimulus out there, so it's not a strong response like a real stimulus. This is a real stimulus. The neuron actually responds much better to a real stimulus than just sort of imagining a stimulus. But this probably represents some kind of, imagery. And so you could see these influences from different modalities kind of percolating back, because presumably if you went to somatosensory cortex, when the animal touched this bar, you'd see a huge response because somatosensory cortex is interested in touch. But some of that information kind of percolates into the visual system, percolates back into the visual system, and probably activates some neurons to sort of prep for the actual, stimulus. So I thought all in all it was a very interesting experiment. And one last thing, they didn't find any neurons that showed modulation in response to this touch until the animal learned the real task. So they were looking for them when the animal was doing the fake task, and there were no neurons. And so after 3 months, you found neurons that modulated their response depending on what, visual neurons that modulated their response depending on what you touched. But it didn't actually appear until the animal was doing the actual task. Yeah, question?
And they're only touching it, they can't see it?
They can't see it, yeah. They can't see it, they're just touching it, yeah.
But how is that eliminated then? They can see it from the imaginary perspective.
Oh, this? Oh, the imagery case? Well, so the idea is that, there's something blocking, and so there's something blocking their visual field, and so they just touch it, and they feel a little vertical, and then these visual neurons become slightly activated, and then real visual things come in, and you get a big burst of response. But again, are we understanding how this works? No. I mean, there's all these different areas. Signals going back and forth. We don't really know. But it's just kind of a little hint that maybe this is what imagery looks like, because you don't want imagery to sort of be as bright as the real thing. Otherwise, you start hallucinating. So you want it to be like, just a little bit. Okay, so if you read in the literature, if you want to read any of these experiments, there's thousands of all different kinds of experiments of this kind with humans, with animals and humans. But these were kind of like some classic experiments, and there's not a lot of variation in how people interpret these experiments. There's thousands of experiments, but so it doesn't hurt to go through just 2. Okay, so attention, all done in one day.